Hello students and welcome to the study portal. The topic of today's video is different laboratory equipments. So in today's video we will cover different laboratory equipments that are used in the laboratories. It will help you to identify the different laboratory equipments. Alright, so with the help of the identification of laboratory equipment, it will be easy for you to walk inside the laboratory. And we will discuss about the different uses of these laboratory equipments, that in what equipment is used for which kind of laboratory test. So let's start the video and learn to identify different laboratory equipments. So first of all, we will talk about the laboratory apparatus that are used. So here in this picture, in the very first slide, you can see the beaker. It is a glass beaker. Beaker, what is beaker? Beaker is generally cylindrical in shape with a flat bottom. Here you can see that it has a flat bottom and it has a protrusion which is helpful for pouring out the solution. Alright, it is a simple container. It is used for stirring, mixing and heating liquids commonly used in the many laboratories. Alright, now second one are the test tubes. Test tubes are the most commonly used equipment in the laboratory. These are also known as a culture tube or sample tube. Test tubes are of different types and are available in different sizes and shapes. Test tubes are, there can be a glass test tube, there can be plastic test tube, alright. So these are very important to hold a small experiment which are used for the investigation processes, alright. Mainly the glass test tubes are used in the hematology laboratory, in the biochemistry laboratories and the microbiology laboratories. Alright, so it consists of a finger-like length or of glass or clear plastic tubing, open at the top with a rounded U-shaped bottom. Next are the Florence flask. Here you can see in this picture, these are the Florence flask. It is named as Florence flask. So Florence flask has a round body. You can see in the picture, it has a round body with a single long neck. It is open from one side at the top and it is round or flat at the bottom. Alright, so it can be used as a container to hold solutions of chemicals. It is designed for uniform heating and ease of swirling or stirring the solutions. It is produced in a number of different glass thickness to stand different types of uses. Alright. Now talking about the triple beam balance. Triple beam balance is an instrument which is used to measure mass very precisely. Alright. It is very precise. It has it can give an error of only plus minus 0.05 gram. Because it is very precise and accurate. As the name refers, triple beam balance to its three beams, where the middle beam is the largest, the far beam of the medium size and the front beam is of the smallest size. So it is used to measure mass very precisely. If you see instrument like this, please identify it as triple beam balance. Now next one are the Erlenmeyer flask. Erlenmeyer flask are also called as conical flask. It is widely used. It is flat at the bottom, has a conical body and a cylindrical neck. As you can see in the picture, the bottom is flat, the body is conical as a cone, as of the shape of a cone, and the neck is cylindrical. It is suitable for heating liquids. It has a small neck. The small neck reduces evaporative losses compared to a beaker, because when we put the solution into the beaker, the evaporation rate increases due to increased surface area of the beaker, because the surface area of the beaker is higher than or larger than the conical flask. Thus, the rate of evaporation is high in beaker but less in the Erlenmeyer flask. Alright, because of the presence of the conical flask, it makes it unlikely to tip over and spill the solution. So this is the advantage of Erlenmeyer flask. Moving on, next you can see on your screen is graduated cylinder. So graduated cylinder are generally the glass cylinders which are of cylindrical shape and has a bottom, flat bottom. Alright, so it is used to accurately measure the volume of a liquid. What does graduated mean? That it has markings of the measurement marked on the cylinders. So it is used to measure the volume of a liquid. So suppose we have to make a solution of different concentration, then we measure the solution in this graduated cylinder. Water displacement can be used to find out the volume of a solid. These are generally more accurate and precise for this purpose than flask and beakers. Now funnel, you can see. So funnel is a pipe with a wide, often conical mouth and a narrow stem. So it is used to channel liquid or fine-grained substances into containers with a small openings. So the containers which have very small opening and we know that if we will transfer the solution, then it will spill somewhere. So if we don't want the spillage of the solution, we use funnel to transfer any kind of solution into narrow mouthed container. So this is what is funnel. Now next one is the Bunsen burner. Bunsen burner is a laboratory instrument that provides single continuous flame. Alright, here you can see in this picture, Bunsen burner are widely used in the clinical pathology laboratory and microbiology laboratory as well. These are used to heat the solution. Alright, so what they do, it provides single continuous flame by mixing gas with air in a controlled fashion. The ratio of gas to air that is mixed together can be manually adjusted by the knob of the burner, the knob which is present in the burner. Alright, so it allows the user to control the intensity, temperature and the size of the flame. Whatever intensity or temperature is required, we can fix in the Bunsen burner. Now these are the reagent bottles that are present in the laboratory, which are used to store the chemicals or the solutions in the laboratory. They are usually made of glass, plastic or the borosilicate. The material of the reagent container depends upon the type of solution or the chemical that is intended to be stored in these bottles. Alright, so there are some solutions that will react to the sunlight or light. So those solutions which will react to the sunlight or the light, they are stored in the amber color bottles. These are the brown color bottles. These are called amber color bottles. So they are stored in the amber glass bottles. Alright, special caps are or stoppers are present in these reagent bottles to minimize the spillage or the reaction of these solutions or chemicals. Alright, so these are stored in the cabinets or the shelves. Now next one you can see the picture is pipette. Pipette is a laboratory tool commonly used in chemistry, biology and medicine to transport a measured volume of liquid, often as a media dispenser also. Alright, so these glass pipettes or plastic pipettes are also available nowadays. So these are the pipettes which are used to measure a small volume of liquid that has to be transferred. So pipette is used for transferring the small volume of liquid. Now these are the micro pipette. They are more precise and accurate. They are used for transfer of volume of very small volumes of liquid in the microliter range. So these are available in single channel and multi-channel variants. Alright, so some pipettes are of fixed range, that it will transfer from 10 microliter to 100 microliter only, and some are not fixed. So these are the micro pipettes which are used to transfer microliters of the solution to perform any kind of test. Next one is the centrifuge. Here you can see the centrifuge. Alright, so centrifuge is a device that works on the centrifugal force. It is generally used to separate the different components present in the liquid. For example, if we put the tube or the vial of the blood into this centrifuge, the different components of the blood will be separated on the basis of the density and the size of the components. Alright, so higher density, higher sized molecules or high density molecules will settle at the bottom, and the lower density molecule will be separated at the top of the glass tube. So this is achieved by spinning the fluid at high speed within a container. Thereby it will separate the fluids of different density, that such as cream from milk or liquids from solids. So for separating the components in a fluid, centrifuge is used. This is a centrifuge machine in which the different containers are put and it is balanced. It works on the centrifugal force. Centrifugal force works on the liquid, and due to different density the components are separated. Moving on, now talking about water bath. So water bath is a laboratory equipment which is needed to heat the fluid or any kind of solution, as well for incubating the incubating the liquid or the solutions. So for example, if any test requires a certain temperature to reach, then we put the tubes or glass test tubes or the solutions into the water bath so that the temperature will be carried out. So for reaching the different temperature, water bath is used. Alright, so it is preferred heat source for heating flammable chemicals instead of an open flame to prevent ignition, because in open flame if the flammable chemicals will ignite and will produce heat. So it is also used to incubate samples in water at a constant temperature over a longer period of time. Moving on, next is the cell counter. Cell counter is generally used in the hematology laboratory for counting the different types of cell inside the blood, such as the RBCs, WBCs, the different types of WBCs for giving the DLC, and the platelet count. Alright, so it is an important subset of cytometry with applications in research and clinical practice. So this is a cell counter which works on the electrical impedance principle and counts the cells of the blood. Next one is the incubator. So incubator is a device which is used to grow and maintain microbiological cultures or the cell culture. As the name implies, incubator. So it is used to incubate the culture media or the media which is used for growing the bacteria or providing a specific set of temperature, humidity and other conditions. Alright, so it is most commonly used temperature incubator for the frequently used bacteria. Now next one is the hot air oven. Here you can see the hot air oven. So hot air oven are the electrical instruments which are used to provide sterilization. Alright, so as we covered in the sterilization video, that there are two types of sterilization through which the substances in the hospital or laboratory can be sterilized. The two types of sterilization are, first one is the dry heat sterilization and second one is the moist heat sterilization. So to disinfect and sterilize the glass or the glass tubes or the glass equipments, the hot air oven is used to disinfect these materials. Whatever we are using, whatever equipments we are using in the laboratory, they need to be sterilized before performing other tests. So these are these occurs with the help of a hot air oven. It is electrical device. It works on the principle of dry heat sterilization. It has double walled insulation that keeps the heat in and conserves energy. The inner layer is a poor conductor and the outer layer is metallic, which is good conductor of heat. Thus it works as the insulated wall and the temperature is maintained inside the instrument. Now second one is the laminar air flow. The laminar air flow is used in the microbiology laboratory. This is a cabinet or tissue culture hood. It is carefully enclosed bench which is designed to prevent contamination of the bacteria or various pathogenic organisms into the environment. Alright, so air is drawn through inside the laminar airflow through a HEPA filter and blown out in a very smooth laminar flow towards the user. So it comes into different stages for providing different types of protection. Next one is the autoclave. So as I told you that there are two types of sterilization which are performed in the laboratory. First one is the dry heat sterilization and second one is the moist heat sterilization. So for culture media we cannot use hot air oven because it will burn the media and resulting in the denaturation of the media. So for the materials which cannot be sterilized with hot air oven, we use autoclave. It is a machine or an instrument that works on the principle of steam under pressure to kill harmful bacteria, viruses, fungus and spores on the items that are placed inside its vessel. So items which are put inside the autoclave are heated to an appropriate sterilization temperature. It generally works on the temperature of 121 degrees Celsius for at least 30 minutes by using saturated steam under at least 15 psi of pressure. Alright, so for performing different sterilization of different items, different temperature and different time is used. Alright, so it is used for the steam or the moist heat sterilization. Next one is the microscope. We all are aware of the microscope. So microscope is used to examine small objects that are not seen with the naked eye, that can only be seen with the help of a lens, magnifying lens. So magnifying lenses are used in the microscope to magnify the smaller objects such as bacteria, cells or various items. Alright, so the lens bends light towards the eye and makes an object appear larger than it actually is. So these are the different components of the microscope. In the picture you can see there are different components of the microscope are labeled, which is eyepiece, nose piece, high power objective, low power objective. Alright, so generally the microscope comes with the 3 power objectives: lower power objective 10x, 40x and the higher power objective 100x. 100x works as oil immersion lens. For this cedar wood oil is used. Next is the volumetric flask. Volumetric flask is a type of laboratory apparatus which contains a precise volume which is used to calibrate a precise volume at a certain temperature. They are used for precise dilutions and preparation of standard solution. As you can see in the picture, there are different types of volumetric flasks. Alright, so these are also used for making up solutions to a known volume that we know. Next one is the laboratory thermometers. Laboratory thermometers are used to measure temperature. Alright, there are many types of thermometers used in the laboratory, such as differential, mechanical, logging, etc. So they are progressively providing digital reading displays and are input capable to computer and software programs for logging process. It is used to measure the temperature inside the laboratory. Next one is the analytical balance. Analytical balance is also called as lab balance. It is a class of balance designed to measure small mass in the sub-milligram range. For example, we have to prepare the culture media. So for preparing the culture media, the powdered substances are measured in the analytical balance. It is usually include draught-proof weighing chambers for precise measuring, and often used alongside anti-vibration tables to increase accuracy. Next is the mortar and pestle. Mortar and pestle are used inside the laboratory to crush or ground the substances to make the solution. In solid state chemistry, a mortar and pestle is often used to prepare reactants for a solid state synthesis. Alright, so for the ceramic method, the mortar and pestle are generally used. It is used to crush, mash or grind materials or substances in a mortar. So that's all about the laboratory equipments. These were the common equipments or the instruments that are generally used inside the laboratory. Hope you will be able to identify the different types of equipment and instrument in laboratory, and you are aware with the use of all these types of instrument and equipments. Keep learning, keep studying. Thank you.